Hello everybody, this is Mr. Burke. This is my first online screen recording full lesson, so please forgive me if this doesn't go perfectly well. We're all learning here, and especially the teachers are really trying hard to figure out how to convert our lessons into digital lessons. So I'm going to do my best today. Um, Here we're going to start with 3.3. First thing that I always try to do with IB Bio is have a look at the presentations. So I think it's really important to look at the statements and make sure you read through them and try to understand what they mean and try to flag any that maybe specifically seem really confusing to you. So I would just maybe pause the video, read through these. You could also have your presentation up with you and just see what looks familiar, what doesn't. And then, of course, I think that the guidance is really important to look at as well in terms of it provides a lot of information as how far you should actually go into the material. So it can be really useful to read those sometimes. Um, and then if we go, these are all the understandings, right? The things you need to know. And then we go into applications and skills. So these are the things that you actually have to be able to apply. So you're applying this knowledge of non-disjunction can cause Down syndrome and other chromosome abnormalities. Remember in class, I think I mentioned that we would talk about what causes Down syndrome. And in fact, it is something called non-disjunction, where the chromosomes are not separating properly um, after metaphase, so during anaphase. So you'll read through these. You can also look at the skills, obviously. Right here, these are the things you have to be able to do. So you will have to be able to draw diagrams to show the stages of meiosis resulting in formation of 4 haploid cells. Haploid. So hopefully you remember some of this vocabulary. Haploid, I really tried to stress that before. Okay, so first thing I want to do is review a little bit of 1.6 because mitosis is very much related to meiosis, and I think it's important to remember this acronym PMAT, right? And if you can't remember PMAT, maybe you can remember people, meet, and talk to help you remember the stages of mitosis. These are also the stages of meiosis, but they happen what? They happen twice, right? So instead of just prophase, metaphase, anaphase, telophase, it's prophase 1, metaphase 1, anaphase 1, telophase 1, and then we go into prophase 2, metaphase 2, anaphase 2, telophase 2. Okay, so here is an image showing you the structure of a chromosome, okay? And as you can see with this chromosome, it's actually replicated. You can see it's replicated. Why? Because it has that X shape, right? It has the two replicated sister what? These are called sister chromatids, okay? Or you can call them chromatids. Okay, they are exact copies of each other. So if you think back to when we talked about DNA replication, this is the end product. This is what a replicated chromosome looks like. And of course, you know that humans have how many chromosomes total? We have 46 chromosomes. And how many pairs of these chromosomes do we have? We have 23 pairs of the chromosomes. And therefore, we have 46 chromosomes total. This image is actually of just one chromosome. It may look like two, but when you see them connected like this means it's just a replicated chromosome. They're exact copies of each other. This is considered one chromosome. If you were to separate those two chromatids, as does happen in mitosis and meiosis, they become different. They become two chromosomes. This is a very important concept to remember, that it can be very confusing for students. So again, if you have the one chromosome, right? One chromosome, okay? That is one chromosome. If we end up with this structure, this means that the chromosome has been replicated, okay? And we also have a centromere region, which is in the middle, right? There's also something called a kinetochore, which I'll draw in a different color. There's something called a kinetochore in the centromere region, which is a protein in the centromere region that links to the microtubules during mitosis or meiosis. And why does it have to link to those microtubules? Well, because it has to be separated and pulled apart, right? And so this, as I was saying before, is also considered one chromosome. It's just that it is replicated. Okay? If we were to pull those two sister chromatids apart, then they do become two separate chromosomes. Okay, so just something to keep in mind as we're doing this, because the numbers might get confusing. Okay, so back to here, this is what I was talking about with the kinetochore. It's the protein that the microtubules would be attaching to, okay, to pull the replicated chromosomes apart. All right, so if we use this presentation and go to this slide right here, you'll find this nice animation for mitosis. Again, we're going to review mitosis right now. So I'm just going to go ahead and play this so you can remember what mitosis is all about.
Mitosis is a process of nuclear division by which replicated copies of a cell's DNA are organized into chromosomes. The identical copies of the DNA are then divided equally between two daughter cells. Early in mitosis.
By the way, what is the phase in the cell cycle that is before prophase? If you said interphase, you would be correct. And then we're moving into prophase. Remember, in interphase, the DNA is not condensed. It's in what form? If you said chromatin, you would be correct. That's the uncondensed form, which is sort of looks like spaghetti or a ball of yarn or something like that. Okay, so now we're going to move into prophase.
During prophase, the nuclear membrane and nucleolus begin to break down.
Okay, so remember that the nucleolus and nuclear membrane breaks down. I don't know if you remember, I think we did like a hand motion thing that goes with all of this. You can practice that. This is the chromatin that I was just talking about, uncondensed DNA.
As prophase continues, the DNA condenses to form chromosomes, while the mitotic spindle, made of proteins called microtubules, begins to form and attaches to the kinetochores on each chromosome. During metaphase, the chromosomes are moved to the central plate of the cell by the spindle microtubules. In anaphase, the microtubules attached to the kinetochores of each chromosome shorten, drawing the chromatids of each chromosome to opposite ends of the cell.
Okay, So let's back up a little bit, back to this point. Remember the chromosomes are in what shape right here? They're in an X shape. The X shape indicates what about the chromosomes? It indicates that the chromosomes are replicated, right? And so these are actually exact copies of each other. These are the sister chromatids that are attached to each other at the centromere. The microtubules are coming out, attaching to the what? The kinetochore, and then pulling them apart to create two separate cells.
Microtubules attached to the kinetochores of each chromosome shorten, drawing the chromatids of each chromosome to opposite ends of the cell.
So if you can imagine that this is a human cell going through mitosis, which, remember, the purpose of mitosis is to do what? To create exact copies, right? Exact copies of the cells. So you can think of, for example, if you get a cut or some of your cells die, they need to be replaced. They need to make copies of each other to replace the old cells. Also, if you're growing and developing, right? If you're a baby, there's a lot of growth and development happening. This is where mitosis comes into play as well. And so you can imagine if there are 46 chromosomes, right? 46 chromosomes lined up right here in the center, and they're replicated because DNA replication occurred, and then they're separated and pulled apart to two separate cells.
In anaphase, while the unattached microtubules elongate, stretching the cell. During telophase, the chromatids, now called chromosomes, reach the poles of the cell as the unattached microtubules continue to elongate. The nuclear membranes begin to reform as the nucleoli reappear and the chromosomes relax as the DNA unwinds. Telophase is followed by cytokinesis, or division of the cytoplasm. In animal cells, a cleavage furrow forms at the center on either side of the cell, pinching through until the original cell is divided into two daughter cells. In plant cells, cytokinesis is different due to the presence of a rigid cell wall.
Okay, so if we come back to the whiteboard, we can draw this out. So here we have mitosis. It would be starting with these replicated chromosomes. I'm just going to draw two of them. These are the replicated chromosomes. Okay, they're going to separate out. To create the two identical daughter cells. They may not look identical to the parent cell, but they are. They're considered identical because remember, this was just one chromosome replicated. This is another replicated chromosome. They got split up and became two separate chromosomes. And so we do still have the same number right there. Okay, here we call it diploid. And remember, diploid we represent with 2n. In the end, we have two 2n cells that were created. Okay, so exact copies. Now we're going to go through meiosis next. My video is about to run out of time, so I will start a new one and we'll start it on there. Thanks.